Yeah, usually people show up like 5 minutes after.
Totally.
But pretty good seminar.
Usually that's me as well, but.
Good. We're here. Give it like 1 more minute. I think that makes the most. I think that's what your seminar will be. Like an hour. That's not true. Yeah, that's not
Yeah.
But then I think the like side first stuff like that, it gets like a little bit
Right.
Yeah. On your side is like.
I mean, here. I think it's a really good idea, but it's just. We're good? Yeah. Well done. All right. All right. Thank you, everybody, for showing up for this impromptu seminar. I know a lot of us are thinking about spatial transcriptomics and looking at RNA and tissue, and we have Suzanne Mertens from ACD Bio, who is, the company's been developing methods for looking at RNA in situ using fluorescent and other types of probes. And it's a method that we've been using, and Suzanne and I have been talking over the last little while, and we thought giving a seminar here would be a good way to kind of highlight the technique and you know, think about using RNA scope, which is the method that ACD Bio has been developing for, you know, everybody's studies. We find it really powerful. I guess Suzanne's going to kind of highlight some of the technology and some of the improvements that have happened over the last little while, and hopefully it could be valuable to everybody here. So thank you, Suzanne, for showing up.
Thank you, sir.
And the floor is yours.
Awesome. And so my name is Suzanne Mertens, and today we're going to talk about, well, here's some contact information for me.
One thing.
Yes.
We have a microphone.
A microphone.
Put that on.
Testing 1, 2. How does that sound? Cool beans. Okay, so what we're going to do is talk about visualizing what we can't see, which in our case is RNA transcripts and gene expression in general. So first off, here's some contact information for me. I'm your ACD sales rep for the Boston area. And so I'm part of a larger group of dedicated application specialists, field scientists, as well as senior product managers. But I'm first here to help understand what y'all want to do and then route you to the right person or resource or technology to make that move forward. I got my bachelor's out of Georgia Tech. Did a significant amount of work in flow cytometry before shifting to gene expression. Spent some quality time as an application scientist for FlowJo, a premier bioinformatics software package for flow, and then some solid years with Sony Corporation as a field scientist for their flow cytometry group. But when I came across an opportunity to look at gene expression in situ, it was like, I like visual, so let's jump into this, where previous genomics analysis was a little too discrete and cut and dry for me. A little bit of a background on the company itself. Bio-Techne is a holding company. We're pretty big. We're traded on NASDAQ. And it's a constellation of sister companies. Most of them are related to protein and protein science. The very first logo at the top of at the 1 o'clock or 12 o'clock position on the clock there, R&D Systems, is probably the subsidiary sister organization that you would know the most about. But that's us. And we're right now the only ones who work in RNA. So how do we differentiate ourselves from the rest of our sister companies? Well, you're going to come to us if you can't get an antibody, or if there is no way to make an antibody for what you want to see. If you're looking at a secreted protein, it might be a little bit easier to look at transcript. And when it comes to spatially mapping, or more importantly, validating RNA-seq information, we're going to be who you're going to work with, because we can generate data at the single cell level to show and validate what you're seeing in terms of your RNA-seq outputs. In fact, for most newcomers to the technology that are reaching out to us, a significant portion of those people are coming to us because their reviewers on their papers are insisting that they show expression in situ. And so that's where we felt like it might be worth extending out to the larger community and moving it up a bit. All right, so here's the technology overview for RNAscope. So how this works is from left to right. Together, we'll pick a target probe. The probe's hybridization to transcript will form the basis for the assembly of a branched DNA structure that'll allow me to pile onto any one instance of your transcript. It'll let me pile on a significant amount of chromogen or fluorophore to give us some signal amplification. The outputs from RNAscope for detection and quantification are either fluorescent or bright field. And there are a variety of different tools and software packages out there to give you the closest quantitation of these signals suitable for your further analysis. There's two primary publications here, and I'm going to walk through them first. The initial one at top, RNAscope, a new technology for in situ, was published in 2012. And then our secondary paper, which I heavily refer to for the automated quantitative analysis of in situ information, that came out in 2013. So let's dig into that first paper. Li-Xiong Wang, fourth author on here, is still with us. And what we put out there was a novel in situ RNA analysis platform for formalin-fixed paraffin-embedded tissue. There was a deliberate strategy to go after the most prevalent, clinically available repository of samples out there. And right out of the gate, what we were able to do is run a multiplex. So this is a cell that's lit up DAPI counterstained for the nucleus, red for beta-actin, green for HPRT1, yellow for RPLO, and then pale blue for PBIB. The idea was in this paper, which is kind of worth digging into if you have some preliminary or fundamental questions about RNAscope, was what they did in the beginning was deliberately compare and contrast RNAscope to clinically validated assays and scoring methods. Because instead of just saying, hey, this is cool, this is great, this is awesome, we said, why don't we go back and make sure it matches what we already know from things like RT-PCR. So I'll break down some of the figures in the paper, and this one is the beginning of our understanding and description of what I'm going to snap forward to, which is this double Z probe design. I'm going to go back to the original figure. So what you're seeing here is the validation of our unique method. When I look at your transcript, I'm actually going to probe for your transcript with two Z's, a left one and a right one. And what this figure is showing is that when we did the initial testing of this method, we were quite confident that unless I get two Z's of a pair to hybridize together to your transcript, without those two Z's together, I don't have the basis for forming the rest of the association of chromogen or fluorophore to signal. So at the top left, you're looking at a full set. So this is a sample that's had both the left and right Z pair applied to it. On the top right, this is unstained control, effectively, no target applied. The left set and the right set on the lower row are showing what would happen if I only got half of an optimal hybridization from either the left probe or the right probe. I really like this paper. This was the first thing I had to read when I came on board, and it laid it out nice and succinctly without too glossy a series of brochures. So what's a Z? Okay, so this is the beginning and the fundamental kernel of everything that RNAscope does. The lower portion of the Z is the site that's specific to the probe you've either asked us to design for you or a probe that you've pulled out of the catalog. And at the top of the Z is a generic structure that together allows me to make something on the cheap in terms of manufacturing, allows me to make something specific to you, but generic enough it's easy for me to assemble and manufacture for you. Now, when you order a probe set from us, it's not actually just one ZZ pair, except for one particular assay. I'll get to that later. What you get from us when you ask for a probe set is a composite. As many as 20 pairs of these double Z probes. And what that allows us to do is build some redundancy into the system. So if you're working with very old sections, or if you're working with RNA quality that's somewhat suboptimal, if I have a composite probe set, there's a better chance of me lining up transcript to a significant enough signal that you can see it. And so, of course, we had to test that. So we started looking at housekeeping genes in 15-year-old FFPE samples. The ground that you see here at the smallest point here at 100x magnification is a single instance of an RNA molecule. For the pretty explanation of how this works is, so let's say here you're going to specify to me a target sequence. And together with my bioinformatics team, we're going to design a probe. Again, target binding at the bottom, generic linker at the top. And how this works is 2 Z pairs come together, hybridize to your sequence, and those 2 Z's together form a binding site for the larger tree structure that's going to be built out through rounds of hybridization. I have 2 Z's. I have the ability for a preamplifier to hybridize to that. Once I have a preamplifier hybridized, I can go through and apply multiple rounds of amps, building out the equivalent of branches on a tree. Once I have those amps hybridized, I can then go ahead and apply chromogen or fluorophore. All that right there is the schematic to show sensitivity, that I can see an instance of your transcript and make it really bright. The reason I have those 2 Z's together is in a goofy situation, either because I have off-target signal, I'm fighting against a significant degree of homology in the rest of your experimental context. If I only have 1 Z bind, well, I don't get anything else to happen. And without anything else to happen, I don't see signal. And that's the basis for me giving you specificity. So if we dig back to that original 2012 paper, it lays out the basis for saying that we are limited. We can only see that which we apply probes for. We are specific. You're seeing HCV infected or uninfected cells. And we're able to do, right out of the gate, a multiplex, a 4-target experiment. The paper also elaborates the quantitative signal molecule detection. Because again and again, we'll get asked, how do you know that you're only seeing 1 molecule of RNA? Well, what we did is we looked at HER2 cells, and we looked at mRNA from mRNA of HER2 in HeLa cells. And we compared that to the numbers of mRNA transcripts that we were able to pull out from a quantagene experiment. The dot number per cell determined by RNA scope exceeded that which was acceptable as concordance with the validation of a clinical assay. And it allows us to extrapolate and make the claim that we can see single molecule detection of RNA. We further poked around into this to bolster that claim by looking at a side-by-side comparison of HeLa cells and SKBR3 cells, looking at an instance of IL-8. In HeLa, you only get 2 copies of it. In SKBR, as a characteristic of that cell line, you'll see multiple copies of that transcript. So this is our way of saying we see only what's actually in your sample. The 2013 paper digs into it even more. At this point now, RNAscope is being run on auto stainers. So if you're working with a LicoBond or a Roche Ventana Discovery, our manual kits are pre-formulated to work on those systems as well. So this is my way of emphasizing to you, this is not an assay that involves a lot of tweaking. It is designed right out of the gate to be automated. And the rest of this paper describes how to look at RNAscope in a quantitative manner and how to pull out data that's not just, well, I can see brown and then I can't see brown. We're quite proud of the fact that between RNAscope and qPCR, we had a 97% concordance with standard fish testing, which means we beat CAP's requirements for what makes for a robust assay in a clinical setting. So for us, that was pretty good. Well, that was 2013, and now we're at 2023. Over 7,000 publications. The data here is from our group as of September 2022. That number is significantly bigger by now. We are in the big journals and have been there for a long time. We've expanded beyond HER2 and support research areas and applications supported by RNAscope technology, from infectious disease, cell therapy, gene therapy, immuno-oncology, cancer, developmental, and neuroscience. And this is a smaller subset of what we can do. I think this will help answer the question for the genomics group in terms of why would you bother with RNAscope? Because can't you just take a lysate and run PCR? Like, yes, you can. You can get massive plexity, but you're going to lose any feeling or sense for where individual cells and their transcripts are next to one another. You're going to get no single cell resolution. Single cell sequencing technology is pushed forward, so now you still have massive plexity. You still don't have spatial context because single cell sequencing requires single cell suspension, dissociation of tissue. But you do get single cell transcripts now, so you're slowly starting to resolve a complex tissue back into its fundamental qualities. But now and today, you get single cell spatial analysis. Now, I can't go massive. I have an upper limit of 48 targets I can hit all at the same time. That 48 target number is also affected by the fixation method of your sample. But I do give you single cell information, and I can give you single cell transcript. Again, here's a walkthrough of the process. It's a unique probe design. It will require your sample to be adhered to an average glass microscope slide, nothing fancy about it. I'll run a permeabilization of your cells or tissue. We'll do hybridization to get probe to bind to your RNA. We'll amplify the signal by applying sequential rounds of hybridization just to the Z pair and the amplifier. We'll visualize with morphology, so you'll be using a scope to interpret your data. And there are a variety of tools out there to quantify single cell expression. But for every other assay we're looking at, for some of you, you have the printout laying out all the different assay classes that we have, but this is the workflow for all of them. We have over 40,000 probes in the catalog. And what you're seeing here is that we go and push far beyond just mouse, human, rat, or non-human primate. If you have an annotated genome, we can generate a probe for you. I'm kind of curious to know what the individual who's studying asparagus needs to do with gene expression, but I don't ask questions around here. I just throw brochures at people. For folks working in salamander, I started going through the catalog, pulling out probes, and then I just sort of lost enough time to pull them all out. But there are more than what you see here on the slide are available for salamander. Zebrafish gets even crazier. This is a small subset of over 2,000 different probes we have for zebrafish. You might notice on any of these that you'll see a gene name and then you'll see a number behind it. That indicates the different variants of that probe set, or the probe set for that gene target that specifies to a particular RNAscope assay. And again, like the zebrafish, we can always generate a new probe for you. Just give us a sequence and have it reference an annotated genome, and we can make magic happen. Okay, so this is the slide I'll come back to as we go through and traverse from left to right the different assay classes that are out there. But this is what we do. If you look at that top row for transcript length, you'll notice to the far left, I have assays that can see long transcripts. But as you go further and further to the right, you see a massive shift from RNAscope to BaseScope. And this indicates where I can use our chemistry to look at shorter and shorter sequences. I can even push it all the way to microRNA scope, where I'm looking at very, very small recognition points between our probes and your transcript. I figured out how to plex a very small target with a longer sequence as well. Go ahead.
How does, you showed the diagram, the two Z's needing 50 and 50 base pairs.
Yep.
How does that work for microRNA that's only 24 nucleotides?
We drop out the Z and we expand the footprint of the recognition sequence and build the branch DNA tree just from that one instance.
So it's just one Z, not two Z's.
And at that point, it's not even a Z. That's part of why the microRNA scope assay class has its own pricing relating to probe design. But whereas previously we were talking about these assays having a composite pool of double Zs, with microRNA, we know we only have one shot to hit that sequence. And we push the technology further now to look at DNA as well. The DNA scope duplex assay, it's a bit of a niche product. It doesn't, it has a significant obstacle in that you only get usually 2 copies of a target in a cell, but you can use the DNA scope duplex assay to do things like break apart assays. Below here, under the row for maximum targets, you can see as we go from left to right, we're able to simultaneously report out a greater and greater number of transcripts. The RNA scope high-plex assay here with this big gamish of icons is indicating to you that depending on the fixation type of your material, I can look at 12 targets at the same time, or I could push it all the way out to 48. When we go to base scope and base scope duplex, I can't go beyond 2 targets. The readout will always be chromogenic. For microRNA, it's chromogenic as well. But once we get into a duplex of microRNA and full-length RNA, then I can start working within a fluorescent readout again. And DNA scope duplex is chromogenic. That is how it is. If you want to get clever with it, your cheapest entry point into RNA scope is to work with the red kit. The red chromogen, you can see by bright field, and then you can also take your slide and image it on a fluorescent microscope as well. It's kind of like the gateway assay to the rest of the powered RNA scope. Brown or red will deposit chromogen. It's not going to fade, it's not going to evaporate, you'll always have those sections forever. Once you're comfortable with the technology, often then customers will shift to the assays that allow for a larger number of targets to be illuminated at the same time. Down here, there's a row indicating channel. When you and I work together to design your assay, we'll be looking at things in the catalog, and that's where the C1 or T1 or S1 will become relevant. It's not really essential to know right now. For all of these assays, with the exception of high-plex and base scope duplex, we can run these on auto stainers as well. So these are assays that are robust enough, I can get a robot to do it for me. For all assays, with the exception of RNA scope plus or DNA scope, these are the assays that I can combine together with IHC. So if you do have an antibody that works, and you also want to incorporate them looking at targets that you can't use an antibody to define, you can use our technology in coordination with an established IHC protocol that you already have. So, yes.
Another question. Is chromogenic qualitative, and is fluorescence the only one that's quantitative?
Other way around. It's a lot easier to quantitate chromogenic. With fluorescent, you have to be really strict at how you do your imaging and how you control for exposure. When you're ready to dig in the technical details of data analysis, that's actually one of the best ways I've found to introduce people to the technology, except the brochure is super heavy and I didn't write 20 of them with me. Let's you and I flip through the guide for post-analysis quantitation, and you'll see what we have there from the various software application packages I have to do quantitation.
How come there's the microRNA assay that is chromogenic, but then the next one over is fluorescent?
Magic. We use.
Like, why can I not get microRNA scope in fluorescence?
If I had a nickel, Every time that it's been requested of me, I could stop working. And that, I don't have a discrete answer to right now.
So I could have the same target, pick a random other RNA and get it in fluorescent, but not. No.
That was our first question as well. For some microRNA targets that have a high degree of expression, I can do the same thing with microRNA scope as I do with RNA scope, which is go for red and just image it on a fluorescent scope.
That's another question, and you may get to this eventually. So we also work with metals instead of colored fluorophores. Are you going to talk about that today? If so, I can wait, but if not, can you comment on it? Because
That question exceeds my current scope of knowledge.
Okay.
But let me share with you some information about another investigator that is working to swap out chromogen or fluorophore with heavy metal.
Yeah, the Standard Bio is already advertising that you do it,
Well, good for them.
but it sounds like you don't quite yet.
No, not that I have in something that's a discrete catalog number. Any other questions? All right. So how about let's just think about getting started. When you want to work in RNA scope, I'll push you to go to the catalog first. See if what you want is already something that we've designed a probe for. Because talking to us is easy to do, if you don't see what you want in the catalog, my knee-jerk reaction response to you will be to open up a request to us to design a new probe for you and start the feasibility discussion based on your target type, RNA or DNA, the type of application, the target name of your gene, and then the nucleotide accession number for your transcript. This compiling of information begins a dialogue between you, me, and probe design to see if what you want to do is actually feasible before we ever get around to ordering anything. So if you have an idea for a transcript and want to see if we have a probe for it, I'm going to push you to the probe design page on the website, and we can start the dialogue about, is what you're looking for feasible with any technology that we have? We can have this conversation before committing to any sort of financial decision. We can get pretty elaborate with it. The form itself is many pages long, and what you can do is dictate. You can say, look, this is the species of my target, but the species of my actual sample is different. So you can look at xenograft in this technology. A little bit of a behind the scenes on how probe design works. You push to us a gene of interest by an accession number. We have our own proprietary design algorithm. To a limited extent, I can give you back some details as to the coordinates of the recognition sites. I can't give you too much granularity without having to sign a bunch of paperwork. But on average, we're going to make a standard probe design, which is going to be a composite 20 probe pair. It'll be looking at about 1,000 nucleotides in all. As some folks will find, they'll dictate to us that, hey, I wanted something in RNA scope, but I think base scope is the only way we can do this. And the reason why probe design might give you a reason to shift to a different assay class is if we can't get a composite of 20 probes, 20 Z sets, we might have to shift down, after we get down to like 5 Zs, I might have to shift you to a different assay class. If you're working with us for the very first time, the intro pack will be both the most expensive catalog number you'll ever buy, and it'll be the last time you order that catalog number. For a totally new user, what we pulled together is a compilation of the pen you need to draw your circles around your microscope slides, the reagent kit for the assay that's the catalog number you'll probably be ordering again and again. But in our intro packs, we also want to make sure that we push to you a defined tissue sample type, as well as a defined probe set that we know works on our side. So the thinking behind this is that without regard to the intricacies of your assay, we're going to make sure that we're also sending you material that will work. And if we see struggles with readouts from the material that we send to you, that lets us know that extra eyes need to be applied to you in a particular experimental context to get past any technical obstacles at this time. The workflows are fully documented. That QR code right there will push to one of our manual assay workflow webinars. For any one of our assays, there's going to be a series of checklists, a quick start guide, as well as the full user's manual. And all of these documents are available from our website at any time. The essential piece of equipment, the thing that you have to have in order to run our assays, is this hybridization oven. If you're not using it, I know there's an alternate vendor, or I know you might have, what I sometimes hear from customers is that they have their own hybridization oven. I'm like, that's cool, but you have to use this, otherwise support won't work with us when we have feedback or troubleshooting that we need to do. The oven itself can be a bit of a rate limiting step for folks, so I do what I can to help new customers find an oven nearby in order to run their assay.
Is there an oven here?
Yes,
We have.
You have one? Okay.
at least 2 that I know of. The oven has 2 temperatures, 40 and 60. And that's it. 60 is for baking the paraffin slides. 40 is the condition we use to backfit all of other probe and hybridization conditions to one temperature. Going further into assay documentation, this was the guide actually I was referring you to talk about how to do your quantitation after the fact. But we've got all sorts of documentation all over the place. Remember, like, a significant part of our portfolio is packaged and labeled for diagnostic reporting. So we've got a lot of structure around what we give both the clinical market as well as to the research market. Okay, so now let's traverse that whole big smear of different assay classes, and we'll break away into what goes on or what you can see with, let's start with RNAscope and RNAscope duplex. So with red and green chromogenic, you can look at tumor infiltrating lymphocytes, you can look at macrophages, you can figure out what subset you're working with in a section. With brown chromogenic, you can look at species-specific gene expression, even in a xenograft model. With red chromogenic, you can look at viral transcript. For long non-coding RNAs.
How do you quantitate, like a 2-fold difference or a 3-fold difference? How do you quantitate from these brown images?
I learned this phrase the other day and it's really helped my life. Your question exceeds the scope of my knowledge, but I know the person we can talk to.
Yeah, there's a programming QPath that makes it very doable. It'll enumerate the spots from samples.
Okay.
And we have the webinar and the tech note describing using QPath for this method. I can share the links to that after this talk. For the pharma crowd, a lot of our business is generated by individuals working in gene therapy that want to use RNAscope both to confirm the promoter, the transgene, as well as the native transcript. So a huge amount of our work, not on the research side, on the pharma side, heavily uses RNAscope in the context of gene therapy. So let's scooch over from RNAscope into multiplex. At this point here, we stay in fluorescent. We can also combine RNAscope targets with immunofluorescence. So I can show you what's a CD8, what's a CD4, and where are my Tregs, as well as who's expressing interferon gamma. And we can see long non-coding RNA in a duplex, or in this place, a triplex. We push the technology further. Actually, this is a funny one. There was a lab at the NIH that hacked the multiplex V2 assay, and they had their own homebrew for what I'm about to describe, which is high-plex. And so there was a group out there that wanted to look at more than 4 transcripts on the same section. And they came back to us and they said, hey, oh, by the way, depending on how you do your probes, your developing and your stripping, we can get as close to 48 targets as most people are comfortable working with. What you have here is expression of genes that I cannot pretend I know their functional significance in this section, but I'm more than happy to share with you the original citation. The high-plex technology, although it can give you a 48 target readout, the imaging for it does not require 48 discrete channels of color. It is a segmented workflow where what you acquire can be recomposited into a single figure with 1 or 2 targets as needed. The workflow for high-plex kind of reveals the secret behind it. For high-plex, to look at 12 targets or more on a single section, we go through iterative rounds of imaging, of developing of the first 3 or first 4 targets, 3 or 4 depending on the nature of your imaging system. So we're basically reusing the FITC channel, Psi3, Psi5, and for folks that have the far red channel, the equivalent to Psi7. This is not a quick assay. To get a full readout of 48 targets, you're looking at a solid week's worth of processing and imaging. Fresh frozen, fixed frozen, and FFPE works as well, but you're not able to do more than 12. All right, let's break away from long transcript RNA-scope and let's look at assays for when your transcript is shorter than what RNA-scope can handle. And so that's going to be base scope. So with RNA-scope, you had about 20 Z's in a composite pool. With base scope, you're working with between 1 ZZ pair up to 3 ZZ pairs. It's a neat way of looking at exon junctions or splice variants. If you're giving us a target that has a high degree of homology to the rest of a genome, base scope is our second round of trying to optimize a system for you that's going to minimize off-target hybridization. But if you're looking for small changes or small variations between target and off-target, between your experimental or your control, base scope is going to be our first pass at looking at a shorter transcript. We can even use it in some situations to look at isoforms. You may see some investigators out there that have used base scope to do point mutation. We have a limited number of probes that are validated for use to detect point mutation. The original literature for base scope gave this sense that you could design a probe against any point mutation. That's not actually how it works. So let's shift away from base scope into even smaller miRNA scope. And this is where customers are asking to look at, hey, I want to see some siRNA. I want to look at an antisense oligo. Maybe I want to just look at a really short RNA sequence. So we're like, okay, so we'll still do a probe design. It's still going to adhere to the 40-degree rules around that hybridization oven. We can make it so it's compatible with protein detection, and it will work with FFPE, fresh or fixed frozen, or cultured cells. You're going to get a readout that's fast red, so chromogenic, and if the expression is high enough, you could do a fluorescent readout on it. And it's robust enough, you can run it on an auto stainer. Let's see, so this is MIR 124-3P. We see its expression in the cerebellum where we're expecting it and not in the liver or kidney. So this is our way of saying to you, we can give you high sensitivity and tissue specificity as well. We can look at various tissues and various ages of tissues. For microRNA, unlike the other RNA-scope assays out there, what we present as a negative control is not an off-target sequence or a sequence to a gene in a species that shouldn't be present in your sample. For microRNA, we just take your sequence and we scramble it. But we can look at cartilage, And we can get down to, in some say, we can get to single cell resolution, and for some individuals in some contexts, we can even get to subcellular localization. We work a lot with retinal samples. But we're not limited to any particular tissue. In fact, we have a whole series of guidance documents on how to do your assay optimization for lung, intestine, kidney, or brain, in addition to other tissue types. For a couple of antisense oligo options, as well as siRNA applications, we can verify that you've got delivery, and we can verify that we've got target going to where it needs to be. So for the microRNA assay, we can look at transcripts that are between 17 to 50 nucleotides in length. You got improved detection of short transcripts, and we can merge together our ISH with IHC. Scooching over to RNAscope Plus, where we're plexing RNA and microRNA together. We've got a variety of different needs coming to us from investigators. And so we thought, let's merge it together. And so let's look at an siRNA along with, let's see, what's this one? A PBS controller at treatment. We can even see and give you a readout for knockdown. So this forms a basis for me throwing down that brochure on the chair there, which was that microRNA with RNA together is the latest product offering that we have. And then a couple minutes to talk about DNAscope, which is a little bit more of an esoteric assay for us. The majority of investigators aren't looking at DNA. But RNAscope and Bayescope share the same probe design. MicroRNA, as I've mentioned already, has a vastly different recognition point. DNAscope is, the best way I have to describe it is that it's Bayescope ported to DNA as opposed to RNA. So one probe, one double Z pair. In situ for detecting target RNA. If you want to validate your PCR, if you're looking at viral integration, or if you just want to look at fish. Or do it chromogenic. Now, we talk about how it's a pain in the butt to do this, but I'm going to skip to the good stuff. Which is that we can merge together protein and RNA together. We call it co-detection. We have two different ways of letting you see protein and transcript at the same time. The first pass through is just to do sequential. So you'll execute an RNAscope experiment, and at the end, you'll do your antibody staining. In situations where we know or see evidence of your antibody epitope, your recognition epitope is not surviving the protease step required for RNAscope, what we do is we shift from a sequential method to an integrated method, which means that we'll go for your primary antibody first, fix, execute your RNAscope, and then come back for the secondary. It allows us to take one of your antibodies and compare it to what you'll see with RNA expression, but it also allows you to see your readout both from what you're used to with an antibody and then what you're getting into with an RNA probe. Or in some situations, it lets you throw on an antibody that can't be used in the context of other probes. For cytotoxic T lymphocytes, that means by immunofluorescence, I can work with CD8, and by RNA, I'll look at gamma and granzyme. There are a slew of papers. I have a endlessly updating spreadsheet with recent publications. I'm more than happy to share that information with you. But in summary, we have a technology that's specific, it's sensitive, you can multiplex it, you can look at splice variants, and you can combine it with IHC. And that's the end of that. Thank you for your time and attention, and if you'd like to get in touch with me, there you go.
All right, thank you, Suzanne. You guys have any questions for Suzanne?
What do you got?
How does this technology already still compare to Stellaris probes and pricing? Like, what's, am I gonna be able to afford this experiment? Like a lot of it.
We're cheaper at the cost of a reduced plexity. So with our most simplistic kit, a brand new person who has access to someone else's oven can crank out single cell expression of a target for well under 5K for 20 slides. As far as anyone's described to me, that pricing is not what you'll ever see with any of the other competitors out there, Stellaris included.
5K. How many genes targeting is that?
The most simplistic assay will drop below 5K, it'll go below 3K.